Yes, sir. Okay, sir.
Yeah, so if anyone would want to answer, you can raise your hand. Vishwajit, Rajata, Chandrima. Anyone?
Sir, wrong option
Yeah, wrong option.
or
Yeah, yeah. You can
uh,
start with wrong
so
option.
option D, detergent. It is not a physical method.
Okay,
It is a chemical method.
okay. What does a detergent do?
It dip. It disrupt the plasma membrane.
Okay, what is there in a detergent?
Uh, Sorry. What, sir?
No, so you told it's a chemical method and it can disrupt the plasma membrane, right? So
Yeah.
chemically, what is present in a detergent that can disrupt it, that can disrupt the plasma membrane?
Um.
Do you know the chemistry behind it?
No, sir.
Okay, you can think about it in terms of like attracts like, and okay, you can maybe take a minute to think. Meanwhile, we listen to Chandrima. Is it okay?
Okay, sir.
Okay. Yeah, Chandrima.
Sir, option A, alkali treatment.
Okay. Um.
It is also a chemical method.
Hmm.
It is a harsh method actually which we are using the alkali which is causing the lipid saponification
Hmm.
which affects actually this lipid saponification is disturbing that lipid packaging in the cell membrane and affects that cell membrane and breaks that cell membrane.
Okay. So chemically, we are modifying the lipids such that they are no longer lipids
Yes.
upon saponification. Okay. What is saponification? What exactly is the substrate and the product? Are you aware of that?
Generally, it is actually formation of the sodium salt of the fatty acids. It gets precipitated. Actually, it forms the micelles. Those the sodium salts makes the micelles and put the lipid inside it, making a extracting it out.
Okay, there is one thing that's right and one thing that's wrong. So let's say you don't use sodium hydroxide, and if you use potassium hydroxide, does it form a sodium salt? Were you able to hear me, Chandrama?
Yes, sir. Can
Yeah,
you please
yeah.
repeat?
So you told it forms a sodium salt and it forms a micelle. I agree with the micelle part. Now, let's say instead of sodium hydroxide, you use potassium hydroxide. Does it still form a sodium salt?
No, that time it will not form the sodium salt. It should be the potassium one.
Huh. So it would depend on what alkali you use. It's not necessarily NaOH. It could be KOH as well. So just to remember that. Okay. And where exactly do we use alkali treatment? Do we use it with all kinds of cells?
We can't because we are having the cell wall. We are not. We can't use. Actually, we can use it on the animal cells.
On animal cells to lyse them.
Yes.
Hmm. Okay. Okay, that's fine. Okay, do you know about detergents chemically? What they make? Yeah, Vishwajit. Do you were you able to think about how detergents could disrupt cells?
So, sir, lipid can be dissolved in no, um it can dissolve with uh, a detergent solution.
Okay, so when we think about detergents, okay, you mentioned rightly about the dissolving nature. Now, lipids are hydrophobic, right?
Yes, sir.
And detergents have a hydrophilic and a hydrophobic region, both. So detergents are amphipathic. They can interact with both water associated moieties and non-water associated moieties. That would mean detergent can physically interact with the lipids in the cell membrane and pull them. That's how they will disrupt the cell membrane. Was
Yes,
it
sir.
clear? Okay. Chandrama, we'll discuss about this alkali treatment and their relevance in the next slide. I have a slide for that. And Rajata, do you know anything about the other two options? Uh, yeah,
So
yeah, Rajata.
I wasn't there. Can you please repeat
Yeah. I
again?
was just asking if you know anything about the other two options. We have eliminated detergent and alkali treatment. So there are two more options left. So you can choose the wrong option and try to explain why that's
Wrong option.
yeah.
Sorry. So permeabilization.
Okay. What does it mean?
So breaking down the cell wall using detergents or enzymes.
Okay. So do you break down completely or is it an incomplete breakdown? How do you define permeabilization?
No, incompletely. It makes cell membrane more permeable.
Right. So you can allow for exchange of different stuff, including salts or nutrient media. Right? That's what permeabilization would do to the cell. So that leaves us with sonication. Okay, so all of us agree on that. We'll just take a look at the alkali treatment part. So, Chandrama, unlike what we were thinking, alkali treatment will not be very specifically helpful for animal cells. Rather, it is for the plant cells. So, what we would, because the plant cell has this cell wall and these layers consisting of cellulose, hemicellulose, and lignin, which are difficult to get ruptured, we use alkali, like NaOH or KOH, either of it. Now, this would interact with these chemical moieties and it would break them down. That is how alkali treatment is specifically used in lysing cells and not for animal cells because they do not have a cell wall. Was that clear, Chandrima?
Yes, sir.
Huh. Yeah, and like Rajata mentioned about permeabilization, it we just ensure that the cells can take up more and more nutrients from the outside by making the cell membrane permeable. And the two commonly used permeabilizing agents include digitonin and Triton X100. While digitonin can only open up outside the cell, Triton X100 has the ability to open up the nucleus as well, permeabilizing the nucleus as well with respect to animal cells. The next question is the osmotic shock method can be employed in the case of, and four different options are given here: bacterial cells, yeast, mammalian cells, and Pichia. So yeah, we'll take a couple of minutes and then try to start by eliminating the wrong options. அது முடிஞ்சிருச்சு முடிஞ்சிருச்சு முடிஞ்சிருச்சு. Yeah, if anyone who would like to start can define with what the osmotic shock method is. We'll take it from there. Vishwajit, Chandrima, Rajata, anyone? Yeah, Rajata. We can start with what osmosis is and then what osmotic shock is. That will help us define and get to the answer easier. Okay, you can go ahead.
So osmosis is the movement of water molecules through a semi-permeable membrane from a region of lower solute concentration, that is higher water concentration,
Hmm.
to a region of higher solute concentration, which
Hmm.
is lower water concentration.
Right.
Osmotic shock is a technique that involves exposing cells to a sudden change in atmospheric pressure.
Okay. So, what is the reason for this change in atmospheric pressure? How does this atmospheric pressure affect cells? If it
Uh,
has,
I guess they are put the cells are put in a put in the hypotonic solution.
right. So, so by atmosphere you mean the environment in which the cell is present.
Yeah.
Okay, right. Now we've discussed osmotic shock. Okay. Now you were talking about yeah, Chandama, I'll come to you in a minute. You were mentioning about how you need a semi-permeable membrane and the transfer of water. So this would mean that you can't have many membranes. So that is how you should think about this question. So if that is the case, which of these options do you think is incorrect?
I answered, right, sir?
Sorry, can you repeat?
I only need to continue, right?
Yeah. So we just concluded that from what you said, we concluded that for osmotic shock to take place, the membrane should be semi-permeable, and apart from it being semi-permeable, it should not have too many layers. If it has too many layers, then the osmotic shock will not happen. So based on these features, do you think, which of these cells do you think will have these features? Having a semi-permeable membrane and lesser number of membranes.
The wrong option is
Hmm.
bacterial cells.
Hmm.
They have rigid cell wall made of peptidoglycan,
Hmm.
so that provides structural support and
Hmm.
protects the cell from bursting.
Hmm.
Yes.
Okay. Okay. Huh. Yeah, Chandrima.
Sir, in osmotic shock, actually, we are putting the cells in a low salt concentration or higher salt concentration.
Are you?
Due to that, it
Hmm.
is we are happening that osmotic pressure. We are exerting that osmotic pressure due to which that cell gets lysed.
Okay. You told both different variants. So, depending on the solute concentration only, you would either allow the entry of water or exit of water. If the water enters, that is when the cell would grow large in size, and something called as turgor pressure and so on would build up within the cell. But if water exits the cell would shrink, as you see in how in case of grapes turning into raisins. So now you can try to be like correct your answer. Do you think it would enter, or do you think water will enter the cell or water will leave the cell in case of osmotic shock?
The water will enter into that cell.
Okay, so if water will enter into the cell, that means what is the concentration outside? What is the kind of solution that you will keep outside?
Actually, we are giving an hypotonic solution to that, due to which it is getting swelled.
Okay, by hypotonic, you mean the concentration of solute that is surrounding the cells will be lesser than inside.
Yes, yeah.
Okay, so now if the concentration is, oh, okay, right. So based on this, which other option do you think can you eliminate based on the information that you have given and we've discussed with Rajita?
uh, Yeast.
Okay. And would that include Pichia as well? Because Pichia is
Yes,
also hmm,
methylotropic yeast.
right. So that leaves us with mammalian cells as the
Yeah.
only option left. Right. So in osmotic shock, it is caused by sudden change in the salt concentration that will disrupt the number of cell types. And here it is equilibrated to higher osmotic pressure, like 1 molar salt solutions. Rapid exposure to low osmotic pressure causes water to quickly enter the cell. Like we discussed, water will enter the cell, it will increase the internal pressure of the cell, resulting in cell lysis. And this is of limited application except where cell wall is weakened or absent. Like we discussed, the water should be able to enter. Right. The next
Excuse me,
question.
sir.
Yeah, yeah.
Sir, I guess your voice is very low.
Okay, I'll speak a bit louder. Give me a minute. Should I repeat what I was saying?
No, sir, we could hear it, but it was
Okay,
very low. That's it.
okay. I'll be a bit louder next time. Give me a minute. Yeah. Uh, yeah. The next question is a frequency of more than dash kilohertz is required for disruption of cells with sonication. Okay. Now, to be able to answer this question, one should be able to think about the audible range of cells, of the audible range in general. Because if we are able to hear sound of a particular frequency, that means that that frequency isn't disrupting cells or breaking down cells. So first try to start with thinking about what is the audible range, and then we'll come to the right answer here. Okay. Yeah, Chandrima, we'll just take one more minute. Yeah, Chandrima, we'll just take one more minute and then we'll start with discussing the frequency. Yeah, Chandrima, you can start with describing the audible range and then we'll go ahead.
The audible range suppose for the human is 20 hertz to 20,000 hertz.
Hmm. Okay.
It's our
And
audible range.
right. So if that's the case, do you think any value lesser than 20 will be 20,000 hertz will disrupt cells?
Yes, because that between the 20 hertz to 20,000 hertz, we can able to hear, or our body cells can bear those waves, sound waves. But less than 20 hertz or beyond 20,000 hertz, it can't bear those.
Right. I mean, we can't hear that. We are not equipped to hear that. But is the frequency alone a deciding factor, or is the ability to hear alone a deciding factor?
Other than that, our cells is also actually hearing is also one perspective, but when you're giving any wave,
Huh.
actually it is going through our cells also.
Right.
But whatever which is not a, uh hearing cell, it is also a cell. It can't bear those sound waves. So another cells also can't bear, because many of when we are hearing beyond that range or ultrasonic waves, we are also having the health issues. So in that case, the cells are also having a problem in that case.
Huh. So, okay, I agree with one point with respect to waves of higher frequency, but what about waves of lower frequency? So, do we agree that waves of lower frequency, that is what we cannot hear, 20 hertz and below, they do not disturb cells? Do we agree with that? Yeah, Chandrima, do we agree that waves whose frequency is lesser than 20 hertz will not disrupt
Yes,
cells? Okay, fine. Okay, right. Yeah, this is a pretty straightforward question. What we need to know is that this value beyond 20,000 hertz is what we need for destructing, uh, uh, for breaking opening cells. And yeah, so that is about this question. The next question is sonication of cells suspended in liquid medium should be done on ice to prevent. Four options are given here. Option A, osmotic shock. Option B, thermal denaturation. Option C, prevent formation of cavitation bubbles. Option D, inclusion body formation. We can take a couple of minutes and then start with justifying why an option is incorrect.
Just reconnecting. I lost my internet connection. I'll join in a minute.